Hello, guys. Welcome to 13 Universal Learning Beyond Barriers, and this is A-level biology session with Shedro. My name is Shedro. Okay, so we're going to be starting a new series on mitotic cell cycle. Mitosis. Have you heard about it before? Interesting. Okay, this is why people have clones. Okay, identical twins. Of course, we are able to grow and we're able to repair tissues and a lot more. So we're going to be starting series 1. So let's dive into the question. Come with me, guys. Let's go. Get to a writing part. It's time to apply our learned concept. Okay, come on, come on, come on, come on, come on. Let's go. Number 1, for organisms undergoing, these are for organisms, read with me, for organisms undergoing sexual reproduction, a reduction division occurs before fertilization. Which reason explains why this is necessary? All right, why is it necessary, guys? Why? Okay, why is it necessary, guys? Don't forget that reduction division. You know what's called reduction division? This is mitosis. Reduction division is meiosis. You know meiosis? Hmm? Meiosis. You know meiosis? Hmm? Meiosis. Yeah, meiosis. Meiosis halves chromosome number. Okay. Now it halves chromosome number. This is a cell with diploid. Okay. So this is another cell with diploid. What meiosis does? This is a cell. Okay, that's a cell. Now watch this. These cells normally, when they undergo meiosis, their chromosome number is half to haploid, from diploid to haploid. Okay, and so meiosis does this, all right? So that these are gametes. Gametes have haploid cells, so that during fertilization we can restore back the diploid number. But guys, imagine that meiosis did not occur. Let's assume meiosis did not occur. So this was not halved. So that means the gamete of this guy, the man is still having 2n, and the woman, the gamete, the gamete is still 2n. That's haploid. Okay. So what happens is that it's going to double the chromosome number. The offspring will have 4n instead of 2n. Okay. So the implication, this is quite scary, guys. Think about it. It's scary. Now, in every generation, the chromosome number will double if meiosis doesn't play its role. It halves chromosome number. Okay. This means that in every generation, the characteristics of the parent will be doubled in the offspring. So if the parent had, for example, 2 eyes, the offspring will have 4. In the parent had 1 head, the offspring might have 2 heads. That's a monster. That's quite scary. So meiosis saves us from this quagmire. Say, don't worry, I got you. It just halves the chromosome number and then get us back. So the offspring end up having same chromosome number as the parent's organisms, parent cells, parent. Okay. So it's not to increase genetic variation. Okay. Basically, it's not. That is why having chromosome number doesn't create, all right. It might have, it might help because of random fusion, but that's not a major reason. Okay, why it's necessary? All right, because crossing over happens in meiosis. This is above the AS syllabus. Okay, but you know that the major reason is preventing double of chromosome number, reducing chance of mutation. Mutation occurs anytime. The major reason is to prevent, all right, the doubling of chromosome number every generation. So that is 2. Let's go to the next question. What occurs during metaphase of mitosis? Metaphase of mitosis. What occurs then? All right, chromosomes attach to the equator of the spindle. Is that okay? Chromosomes become shorter and thicker. Chromatids reach the poles of the spindle. Each chromosome forms 2 chromatids. So what do you think is the answer? at metaphase, of course, chromosomes line up at the equator. They line up at the equator. Metaphase, that's the most significant feature of metaphase, is that chromosomes line up at the equator. But look at this, chromosomes become shorter and thicker. This is prophase, right? Chromosomes condense, become shorter and thicker. This is for prophase. Since chromatids reach the poles of the spindle, of course, this is happening in anaphase, late anaphase. Okay, they move to the opposite pole. This says each chromosome forms two chromatids. This is also in prophase, right? So this is D and B in prophase features. C is anaphase / early telophase feature, and A is metaphase. Equator is metaphase, right? Chromosome lining up. So the answer is A. All right. Question 3. What are the conditions in a human cell just before the cell enters prophase? Yep. Now, this is very interesting. Just before the cell enters prophase, what are the conditions? Okay. Come with me. Remember that there's what is called cell cycle. After explaining the cell cycle, I will expect you to answer the question, guys. This is cell cycle. So remember, this is mitosis. This is mitosis segment. Okay, don't forget that this is that is cytokinesis, and of course this part is G1, growth phase 1. G1, growth phase 1. Right, this is S phase, and this is G2. Okay, don't forget that when the cell is newly formed, it is in G1, right? That is when cytokinesis has occurred. So the cell is young, and the cell has one ambition. So if you saw the cell and asked the cell, hey guy, little guy, this is a young cell after, say, what do you want to be? You know, the cell will say, I want to be like mama when I grow up. I want to grow big like the mother cell and split for have my own kids. You know, that's what the cell will be telling you. Right. So basically this is interphase in action, right? So you see in G1, the chromosome number of the cell is 2n. Okay. In S phase, that is when the chromosome number doubles. All right, that is when DNA is semi-conservatively replicated. It is still 2n at the beginning. It's towards the end, which is at the beginning of G2. That now chromosome number is doubled. At G2, it is 4n. All right. Mitosis, all through mitosis, it is 4n. And of course, part of this is cytokinesis, 4n until split, cell split. Okay, so with this, let's solve this question, guys. All right, so number of chromatids, of course. All right, just before, look at what I said, just before it enters prophase. That means here, this is G2. Okay, this is mitosis. The first stage of mitosis is prophase. You remember? Remember, the first stage is prophase. So prophase is here. Second is metaphase. Third is anaphase. Fourth is telophase. You get? So just before it enters prophase is G2. So at G2, the chromosome has doubled. Semi-conservative replication must have occurred because it occurs here, so it's doubled. So it's not going to be 46, it's going to be 92. Okay, so if chromatid is 92, then DNA will be 92 because one chromatid contains a DNA molecule and 8 histone protein. Don't forget that. One DNA molecule contains, so one chromatid contains a DNA molecule and 8 histone proteins. So one chromatid gives us one DNA, 92 gives us 92 DNA molecules. Spindle present, not yet, because that's just before, which is at G2. So spin formation, begins at late prophase. Nuclear envelope present, yes. It has not yet disintegrated. That happens in late prophase too. So guys, yes, it's not A, it's not C. If you check, it's not B either. The answer is delta. Delta. All right, chat us up. We want to comment. Let's go on. So each of the following event takes place during mitosis. Okay, look at events. Pause this video and try this out, guys. All right, try this out. This is a sequence of events. Each of the following events takes place during mitosis. The first one says centromere divides. Okay, the question says in which order do the event take place? So we're going to look for the one that has to do with prophase. So prophase first, centromere divides, that's not prophase. Chromatids move to opposite poles, no. Chromosomes line up, no. Chromosomes uncoil. Now this is prophase, right? The two chromatids are joined by a centromere. Yes, two of them are prophase, but which one? No, uncoils. Chromosomes uncoils. This is telophase. Yeah, that's telophase, right? In prophase it coils up, all right? But in prophase, in telophase it uncoils. So prophase is 5. Two chromatids are joined together by a centromere. So go to option that starts with 5. That's the answer. And you follow it. The next is 3. 3 is the chromosomes line up along the equator. That's metaphase. So after prophase, metaphase. 1 says centromere divides. That's happening at anaphase. Okay, that's a late metaphase / early anaphase. Okay. 2 says chromatids move to the opposite pole. That's anaphase. Okay. And then that's prophase, metaphase, anaphase. 4 is telophase. So D is the answer. Okay. D is the answer. The diagram shows the cell cycle when radioactive nucleotides are supplied to dividing cells. This is divide, not driving, it's typo. Dividing cells. At which point will they be incorporated into the chromosomes? Nucleotides. So when are we going to incorporate the nucleotides? Remember that nucleotides are building block for DNA. So the question is, when is DNA made? DNA is made is interphase. Okay, which part of the interphase? S phase. So S phase, if you check here, should be G1. This is S phase. When the DNA replication starts, by the time we get A to A, it's already part of the cell. So A should be the answer. All right, DNA is not synthesized in mitosis, it's not synthesized in cytokinesis, synthesized in interphase, S phase. So this is G2. By G2, these nucleotides are now part of the DNA. So the answer is quite A. C says for organisms undergoing sexual reproduction, erosion division occurs before fertilization. Which reason? I think we'll solve this question. Yeah, what's the answer? Pause it. What's the answer? Of course, only prevent doubling of the chromosome number. All right. See what occurs during metaphase. I think we'll solve this question. You see, they repeat these kind of questions. Okay. Pause the video and answer it. Of course, at metaphase, chromosomes attached to the equator. All right. What are the conditions in a human cell just before the cell enters? We'll answer this. The answer is D. Okay. Which statement is incorrect for mitotic cell division? DNA is replicated. That's it. Now watch. Replication occurs in interphase. Interphase, that's S phase in interphase. S phase is not part of mitosis. Mitosis is not part of interphase. Okay, and it's incorrect. So this is incorrect for this. So A should be the answer. But let's check out B and the rest. DNA is normally unchanged from one generation or cell to yes, it remains genetically identical. That's for mitosis. C says the daughter cells have the potential to produce the same enzymes as parent. Yes, because they have the same genes. Mitosis produces genetically identical daughter cells that have the same genes as the parent cell, so they will make the same protein. Okay, now this is mitosis. D is also mitosis, so it's only A that does not happen in mitosis. The answer is A. All right, so the diagram shows the chromosomes of one cell which has been squashed during mitosis. Which stage of mitosis is shown and what is the haploid number of chromosomes? All right, look at they are aligned at the equator. So is metaphase. There's a haploid. If you count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So that's the diploid. So haploid is 5. Okay? So it's metaphase and haploid is 5. See. Thank you guys. It's raining here. Alright, we're gonna stop now. Please remember to subscribe, leave your questions, share, and of course comment. See you next class. Bye. This is Shedro.